will Gulf of Maine populations evolve in the next century, estimating the evolutionary response of blue mussels to multivariate climate stress. And okay, so as we're all aware in this audience, our oceans are changing potentially quite rapidly as a result of human activities. And it's also important to remember that this is a multivariate change. So we're getting change across several variables that are in general quite stressful to marine organisms. And so we've given you just a screenshot here of some model results of Camille Mora, a paper by Camille Mora published in 2013, illustrating when you integrate across these variables, there are obviously some regions of the ocean that are predicted to be worse than others. And look up in New England and the Gulf of Maine, we're in one of those areas. So we also know from data collected during the last 30 years that the Gulf of Maine is changing very rapidly. And if we just take the last 10 years, for example, we know that rate is pretty much higher than any other place in the world. If this is just one variable now, this is temperature. So the question is, how will populations that we're interested in from either a biodiversity perspective or an economic perspective, how will they respond to that change? And we're interested in that answering that question in a blue mussel system. This is the genus Mytilus. And we're interested in using this system because one is they're a growing aquaculture species in Maine, becoming more popular. They're quite hardy, quite easy to grow. And they're also a great calcification model because, of course, one of those stressors is ocean acidification, declines in pH. We want to know how that stress might affect the ability of a variety of organisms to calcify in the next 100 years. And there is some information from mussels on some of the metabolic pathways that are important to calcification. So in that respect, it's a reasonable calcification model. The way we really think about this is we're interested in how much genetic variation there is in a stress response traits in natural populations. So we can think about the response to climate change being physiological, and there's been quite a lot of work on the physiological aspect of how organisms can deal with stress. The other part of the equation, though, is how is it possible that natural selection could essentially save some of these populations from climate change with existing genetic variation. So that has an important ecosystem implication. Will natural selection rescue natural populations from climate change? But it's also key to aquaculture. Can we identify genetic strains that will essentially be very good at resisting climate change in the future? So the way we think about this quantitatively, it's actually a very old problem in the field of quantitative genetics when we think about quantitative traits, and many of our stress traits are indeed quantitative. They show continuous variation. And if we think then, if we think about the phenotype, some phenotype we're interested in measuring that say is an important trait to resisting climate change. There can be a lot of population variation in that phenotype, and that is a result of various sources within the population. You can have phenotypic variation due to environmental variation. Another way to think about that is phenotypic plasticity. As I mentioned earlier, that's been fairly well studied in a variety of organisms. It's an ongoing area of research. But you can also think about the genetic component, how much genetic variation is there in that phenotypic trait. And you can add complexity. There can be genotype times environment interactions as well, contributing to that overall phenotype. But this is kind of the basic equation that describes phenotypic variation. And traditionally, the way to approach that is to do essentially a set of controlled crosses, so we know who the parents are, and look at the offspring and measure some attributes, either genetic or phenotypic, in the offspring. But we are interested in a method that we can apply to almost any system, including systems that haven't been very good or easy to study traditionally. Either they're difficult to culture, larvae or adults, or they live so long that you can't really phenotype them because you're going to be waiting 50 years or so before you can get that data. So we're kind of interested in a population genetic shortcut to identifying the source of variation, and what we've come up with is a method which essentially uses existing, we call it standing population variation in highly multilocus loci called SNPs, single nucleotide polymorphisms. We can potentially link that variation to some phenotype of interest. In this case, we're talking about a stress phenotype, and we'll define that more clearly for you shortly. And we can also do things like link this interaction to another one, that is how gene expression patterns are driving essentially stress phenotypes, because we know, of course, gene expression has strong genetic components. So in the jargon of quantitative genetics, this is called quantitative trait loci, the ones that can controlling stress phenotype, but we can also think of expression quantitative trait loci that control gene expression, just so we're all on the same page. Okay, so we're going to focus mostly on this interaction today in blue mussels, but we'll also talk, Sarah will also talk a little bit about gene expression patterns too. Okay, so we're working in the Mytilus system, and as some of you might be aware out there that love and study mussels, it's a system of really two closely related species that are very difficult to essentially tell apart with any method besides genetics. And so we're working with essentially samples from each parental species that are basically distinct genetically, and we're also working with some individuals that are mixtures, admix from hybridization of two individuals as well. So it turns out that if in some cases that is an advantage to this problem, because these individuals actually can give us quite a bit of power to associate genotype with phenotype. But it's also true that it's very difficult to separate them anyway, so we sample them all. And our sampling in the Gulf of Maine is pretty extensive. It ranges from the southern Gulf of Maine down here in, thank you, Sarah, all the way to the Bay of Fundy, essentially up the north in Cobscook Bay. And we've got pretty reasonable sample sizes from these sites as well. So we're really trying to cover all the variation we might find in the Gulf of Maine. Okay, so a little bit about the experimental design, how this works. So we essentially measure a phenotype. In this case, our phenotype is the shell calcification rate under physiological stress. So we're growing them under climate change stress. We do this in a common garden stress experiment. We did a 14-day exposure, and we manipulated three things: temperature, pH, and food levels, and we'll talk specifically about those in a minute. After the experiment's over, we sequence every individual in that sample across many SNP loci. In our case, it turns out to be hundreds of thousands. So it's even better than 23andMe. You can think of it that way. And then we utilize a statistical model called the genome-wide association survey, also another acronym here, full of them, GWAS, to actually link the genotype and phenotype. Okay, so the details on that common garden, the stress exposure, are here. So in the control situation, we did a control and an experimental just to compare the response. We have ambient food availability in our flowing seawater system, ambient summer temperature in Casco Bay, 17 degrees Celsius, and then ambient pH is around 8 in that control. And in the treatment, we reduce food availability by essentially filtering out all particles down to something like 5 microns. Thank you, Sarah. We've got a higher temperature, 3 degrees, 20 degrees Celsius, and a lower pH, 7.7, so 0.3 pH units for our climate change control. And you can see we've got reasonable sample sizes in both of these experiments. So what happened that. We do this in our Coastal Study Center, which is a marine lab here at Bowdoin. It's on Harpswell Sound in Casco Bay. We're pretty well set up to do climate change experiments, and this gives you an idea of our setup in the lab. And if you are interested in doing climate change experiments, since I'm the director, please contact me. Okay, so how do we actually measure the phenotype? So basically, we're measuring calcification rates, and we're using a technique that's quite sensitive in measuring calcification changes called buoyant weight. So we basically eliminate the tissue weight and just get shell accumulation over fairly short intervals. So you can actually see changes in a short time periods, a few days. Our SNP, we've got a visual of our SNP assay below. We're doing it using a technique called genotype by sequencing. For those of you that are interested, we end up with millions of short reads that must be aligned, and after we align them and basically figure out which individuals have all loci, we can start to do some analysis. But Sarah will talk about that in more detail. So a little bit more on those methods. As I mentioned, we're assembling millions of short sequences to come up with our loci. We can also measure a variety of population parameters. We can do things like genetic distance, individual ancestry, and we get really nice precision with those estimates of those population parameters because we have so many loci. So it's really a genome-wide survey. And then the last step is to statistically associate stress phenotype and genotype. And it's also important for us to point out that there's no current reference genome for Mytilus edulis or Mytilus trossulus. We are working with a reference genome for the European species Mytilus galliprovincialis, but we have limited resources to actually annotate that, and that's an area that we hope that we'll build in the next 3 or 4 years. Okay, so I'm just going to, my section of talk, I'm going to finish with telling you a bit about the result of the phenotypic experiment, and this has been really, really interesting. And what we found in 2 years that we did this experiment is that when we essentially stress mussels out, growing them at high temperature, low pH, and low food supply, we get a lot more variation in that calcification rate than in the control, and that happened in both years. So we have mussels that are doing quite poorly in terms of growth rate, but we also have mussels that are doing quite well during those conditions. So what we're really doing here is we've been able to express hidden potentially genetic variation by this stress experiment, and that result is also significant if you look at it with a Bartlett's test. So now I'm going to hand it over to Sarah.
All right, so I'll walk you guys through what happens on the genotyping side. So we have our stress phenotype. We see lots of variation. We see increased variation, which is great because now we have this phenotypic framework that we've measured. But what happens with the genotype? So like Dave mentioned earlier, we have this genotype by sequencing assay, and really the goal is to get many, many, many loci over the entirety of the genome. And so far, we've only run a subset of our individuals for this. It's pretty intensive in terms of time and money. So we're actually running the second subset now. So we have results from the first subset, and eventually we'll have a bigger data set. So in the end, we get millions of short reads, and we have to put it back together like a puzzle. Basically, we have to line them all up again. We know which mussels they come from, but we have to reassemble those into interpretable loci, basically. And once we do that, we end up with over 171,000 places in the genome where we can call a genotype. So those are our SNP loci. Once we have that genotype, we can do a few things. So this is just one technique we utilize to try to summarize that genetic variation that we see in the data set. So it's a principal components analysis, otherwise known as a PCA. And so each little dot on this graph is an individual mussel, and they are color-coded by the location in which they were sampled. And this PCA is basically looking at all 171,000 plus loci and trying to distill all of the variation that you see in that genotype into two axes here. So it's really just sort of trying to simplify that very multidimensional genetic space. And what you can see is we have this big split down the middle. So you can see there are some individuals that fall on this right side of the graph and some individuals that fall on this left side of the graph. And that is pretty much our species split, we think, between trossulus over here and edulis over here. And you can say the trossulus are coming from those down east and offshore regions. But there are mixed populations. So Mount Desert Rock and even Cobscoat Bay, some of the mussels sitting right next to each other in the sediment or on the rock are actually different species from one another. And then you can see within those species splits, there is a little bit of differentiation between those geographic locations. So we have lots of genetic variation with which to work. This is just another way to look at that genetic variation, another way to summarize it. Dave referenced 23andMe earlier. Basically, this is sort of 23andMe inference for mussels. So if you look at this graph on the top, basically we have organized our mussels, which are each bar here that you see as a mussel, and they're organized by geographic location on this x-axis. And then the y-axis is proportion of ancestry. So this particular program called Fast Structure, again, looks at that highly multi-locus genotype and tries to infer what the underlying signal is. How many populations are there? So we ran it through many, many different iterations, and it comes out telling us there's two really strong signals, which are, no surprise there, the species. And then we can identify what proportion of ancestry each of these individuals has from those species groups. And so you can see the light blue here is this edulis ancestry, and the dark blue here is this trossulus ancestry. And one thing that jumps out sort of right away is that there aren't a ton of mixed individuals that have sort of what looks like a half and half. We have a lot that have you know, the majority of one species and maybe a little bit of that other, but there are very few sort of very hybrid looking individuals. And then if we pool that signal by population, you can see that geographic pattern. Up here in the north, in Cobscoat Bay, and offshore, we have those trossulus genes showing up, whereas farther south, we have mostly edulis genes. So for that genome-wide association, we use two models that tell us sort of different things. So I want to walk you through what those models tell us and why we use two different ones. So the first model we used is a linear model, and it looks at the data one locus at a time. So it iterates through all the genotypic data for each locus, one locus at a time, and it assesses, is there a link between the genetic pattern we see at that single locus and our phenotype of interest, which is that calcification rate under stress. On the second model that we use, which is a Bayesian model, it can do that. It can do the same thing that linear model does, but it also does a few other things that are really important. It looks at the entire picture, and we can ask how many parts of the genome influence that phenotype. Is it one locus? Is it 17 loci? So that's the number of SNPs. So that's the number of loci that we've measured in that genotype that have something to do statistically with how that calcification rate under stress is expressed. And then we can say something about the heritability. And then we have a really precise definition in quantitative genetics that it's a narrow sense heritability. But that basically just means, is this trait able to be passed from parent to offspring? And we can measure that with our Bayesian model. So to walk you through some of these results, I'll show you the Bayesian model inference first. So I just talked about number of SNPs. That's over here on the left, and heritability, that's over here on the right. And with the Bayesian model, we don't get one answer. I had a committee member for my dissertation that used to call it life with fuzzy edges. So we get a range. So we don't know exactly how many portions of the genome influence that phenotypic calcification rate under stress trait, but we know it's a handful. It's somewhere between 2 and 10. So we get a posterior probability. So somewhere between 2 and 10 loci influence that trait. And then again, we also get this posterior distribution for heritability. the median, mean, mode, they all sort of fall right around 0.3, so that's about 30% of that variation can be explained by genetics, which means it's quite heritable compared to other numbers that appear in the literature when other people measure this in other organisms. That's actually quite high heritability. So we know this trait is heritable. So when we parse out those statistical associations, locus by locus, we come up with 4 loci that have some measure of statistical association with this trait. So here, these are just the numbers that our little program has given them. We can call these locus names. And then we can figure out something about how much each particular locus influences that trait. So higher numbers here are a bigger influence, and smaller numbers are a smaller influence. And then posterior probability is our statistical measure for the Bayesian model, and then the p-value is a statistical measure for the linear model. So what you can see here is we've pulled out 4 of the top associated loci, but we're really only very sure about this one that we've highlighted in red. It has really great association values in both the Bayesian model as well as the linear model. So even though we know there's probably somewhere between 2 and 10 loci, we've identified with great confidence 1, and then we have 3 others that we can investigate. But all of these are anonymous. We don't know what they are. When we put them through the public databases, the Gallus genome, we don't get any hits back. So we don't know where these fall in the muscle genome. So we don't actually know what they do. We just know they're associated with our phenotypic trait of interest. One thing we also did utilizing these models is we wanted to measure our power. Because if you are familiar with GWAS at all in the human literature and medicine, you'll know that we're working really at the low end of what's statistically plausible for number of samples that you can use. So we wanted to assess our power. So we utilized the data we had, and we tried to figure out what our power was, and then what it would be if we had a greater number of individuals in our sample. So our power was about 13.5%, which is low but non-zero. And you can see we did find one really well-supported statistical association. But we have 3 other candidates we can investigate as well. And then along these curves, you can see as we project up in terms of the number of samples that we use, our power increases greatly. So as we get up towards the order of 1,000, we have almost full power. So if we increase our sample size, we can increase power, but it's the cost-benefit analysis too. It's expensive, it's difficult to run these, time-consuming to run these in the lab, so it's sort of, you really want to think if you're going into a system with wild variation, how many samples will I need, or how many samples would I love to do if I had all the money and time in the world, and sort of balance those 2 things. But we have these 4 candidate loci now. And there's potential for using them in aquaculture screening. We could come up with a high throughput screen to figure out if wild mussels have these loci or have these genotypes. But all of these genotypes that are associated with the increased calcification under stress are rare variants. So we don't see them very often, and they're actually all in Mytilus, mostly Mytilus edulis individuals. But they're found in mixed populations, so they're floating around in those mixed populations that you saw earlier with the color-coded circles. So now I want to give you a little bit of hot off the presses. We've talked about the genotype, but my honor student this year did some gene expression work. So I'll talk a little bit about the RNA expression and how that relates to what we've already found out. So RNA-seq is just a technique where you look at all the messenger RNA expressed, and we did this for 24 of the mussels that were part of that stress experiment that we talked about earlier. We wanted to get a big range in terms of what kind of phenotypic variation we saw, so we selected a big geographic range as well as mussels that calcified well and mussels that calcified poorly. And again, we get millions and millions of reads back, and we have to assemble them somehow. But this time, we actually have access to a transcriptome. So my honor student Peter utilized the Gallus provincialis transcriptome. There's actually also from Michael Lesser's lab a edulis transcriptome, to which I'm actually assembling these same reads right now. That analysis is not quite finished. But you can see it's a lot of loci, again, 41,000 transcripts. And then we can test, do these expression profiles look different under climate stress than when the mussels are just happy in ambient heritable sound-like summer conditions. So the results from my honor student's thesis were pretty interesting. So you can see here, I've plotted differential expression. And what that means is on one axis, we have the control individuals. So these were the happy mussels. And on the y-axis here, we have the climate stress mussels. And the dots are all those different loci that I mentioned that were in that transcriptome, those 41,000 loci. And each dot is plotting, what's the expression level here in the control, what's the expression level here in the stress. All these black ones are not significantly different between the two treatments. That's this black cloud that you see behind. And the red ones are ones that were significantly different. So some of these loci have much higher expression in the control, and a handful have higher expression in the climate stress. And another way to look at this is this heat map. So this is really the same information, it's just presented a different way. So the hot colors, these reds and oranges, are high expression, and the whites and creams are low expression. So you can see, again, the control versus climate stress in each locus, each individual transcript is here stacked up on this y-axis. So you can see some of these loci in the control are expressed much greater rate than they are in climate stress. And then there are a few loci that are actually turned on in that climate stress. And some of these are actually heat shock proteins. So we do have some annotation for some of these, but not all of them. So what's the take-home message for this whole story? We know that this trait that we've measured, this calcification under climate stress, is heritable. And there's somewhere between 2 and 10 regions that influence a particular mussel's ability to continue to build that shell under stress, which is going to be important in the future in the Gulf of Maine. And when we see these genotypes that are associated with that ability, they're rare variants in natural populations. We have at least 1 and potentially 4 total loci that we can associate with this climate stress tolerance in wild populations, and hundreds of potential of those EQTL, those genes that are expressed differentially that I just showed you with that heat map, that are either over or under expressed in that climate stress treatment. So I'll hand this back off to Dave here.
Great. So I think overall then that we've shown that we can apply this technique of what we like to call quantitative genetics in the wild to a system that is of interest to a variety of end users, and that there's potential to use this technique on a variety of different kinds of organisms. We have mentioned some statistical and genetic caveats in applying the technique, which really will have to be sorted out a bit for each system. But we think we've really uncovered an approach that has great potential for answering this question about how much variation there are in natural populations that's important to climate change. So with that, we'll also leave off with some future work. The directions we're going is we basically like to develop an assay so we can, a genetic assay so we can screen for these variants in wild populations. And we're also interested in the same phenotypic traits in both in larval and juvenile life history stages of mussels. And we'll just leave you with some acknowledgments. We've got lots of great collaborators. I will mention Floyd Reed, the University of Hawaii, has worked with us on the power analysis. Marco Mondi here at Bowdoin College is to help us set up the experiments, climate change experiments, and a long-term collaboration with Andrew Whitehead at UC Davis has been helpful in thinking about this method. And so I guess we'll take questions now.
Thank you.
All right. Fantastic, Dave and Sarah. Thank you very much. So I'm not seeing any questions in the chat box, and it might be that Jackie needs to tell me how I see those. But in the event that there is or is not, why don't I ask a question that I have? So if you've identified this particular loci, whatever it is, 104243, that is currently, a rare variant out there, have you determined where environmentally it's most prevalent? Is it just randomly distributed across that broad range of environments, or are there particular environments where it's more prevalent, and do you have any idea as to what particular environmental conditions might promote it?
Yeah, I think the answer to that is we don't know yet, because they're so rare that I'm not sure we could answer that very well with the number of individual mussels that we sample that have that variant. But that might be something we can answer with our bigger data set. I'm running 400 and some odd more mussels. So we might be able to then look at those environmental variables. We did measure pH and salinity and water temperature at each site where we sampled the mussels. But I think we just have to wait until we find more of those variants before we can actually answer that question.
Okay, great. And so while you constrained pH, this is my carbonate chemist hat, I'm wondering, do you have any other constraints on the carbonate system? So if it's not hydrogen activity, I mean, do you have any understanding of what the carbonate ion concentrations were, either in the field where you collected the samples and or in the experiments?
Yeah, so we did a titration for total alkalinity, both in our tanks to make sure we knew what our saturation states were in the experimental tanks, as well as in the field where we collected mussels. So we do have omega for both the environmental samples as well as our tanks.
Great.
They're all over the place.
Okay.
But they do fall quite low. They do fall below 1 in the wild.
Yeah. So in the experimental system, I believe we got them below 1.
Yeah, yeah.
So you have them being collected in undersaturated conditions?
Yes.
That's right.
Mm-hmm.
Okay. So, Jackie, I am seeing a list of questions here, but I can't seem to read them on my screen. Is there any way you can read them? I'm only getting half a question.
Sure, I'd be happy to.
I'm not sure. It must be a software issue. Probably you.
No problem.
All right, thanks. I'll leave it to you then.
So the first question is from Daniel McCorkle. So this was at about 10:30. The slide went by quickly, but it appears that the treatment resulted in roughly as many individuals calcifying more rapidly as calcifying more slowly. Did I get that right? Let's see. The same for genomic regions associated with both higher and lower calcification rates. Does it make sense to ask whether the same 4 genomic regions are associated with both higher and lower calcification rates?
Okay, can you see us toggling back?
We can, yes.
Okay. Here's a phenotypic variation, and actually, if you look at those distributions for the treatment here, you see there's like a handful of mussels that are calcifying, like
Super calcifying.
super high in that stressful condition. And there's a whole distribution here that's more towards the middle, and maybe even quite a few that are under.
Yeah, but all the genetic associations are positive, so we don't have any negative. The SNPs are all associated with increased growth and not decreased growth. So the effect sizes would be negative if the statistical model had pulled out loci that were variants that were associated with the decrease. So the rare variants are actually associated with the increased ability.
Yep.
Okay,
That help?
great, thank you. I'm not seeing a follow-up question from Daniel, so I assume that answered his question. Thank you. So next question is from Ron Huber. To what extent are the genotypes in the Gulf of Maine mussels similar to those of more southern regions, southern reaches of the Atlantic?
So that's a good question. We didn't include samples below the Gulf of Maine in our experiments, and actually, if we think about population genetics, I'm not that familiar with the whole distribution across the Atlantic.
Yeah, I don't know that, because this is a anonymous loci, that it's not like they're markers that have been used before. We can't really compare it to mitochondrial sequences or other markers that have been used. Yeah, so I don't really know exactly how similar these would be to more southern mussels.
But we also could add samples of those mussels and use the same genotyping approach, and add it to the analysis for if people were interested either in population structure or in the variation that's down south.
Okay, thank you. So next question is from Janet Nye. What was the reason for having your treatment multiple stressors such as low pH, low food, temperature? Would you have gotten different results if you just tested one?
Yeah, so basically the reason why we're including those is because most of the current work is showing basically interactions among variables. That, for example, you might not get a response to pH unless temperature or food changes. And so initially what we're interested in is, okay, here's an environment that's likely to occur in the future. What kind of phenotypic variation does the organism present to that environment? But you can certainly also start to break it down more and look at single variables or even interactions between variables and look at that response as well. So those are, I guess, questions that you can look at in the future. And you also want to know what you're really kind of working towards is something called a gene regulatory network in controlling this phenotype. And you really want to know how those different stressors are affecting this regulatory network to express, a positive or negative calcification rate. And really that's the most powerful way to look at it, because then you can look at a variety of things that might be changing gene expression through time and through space, which we know is going to be variable. If we think about how our estuary is going to change in the future versus the open ocean, or even different points in the season, we know there's lots of variability out there. So this is kind of our first pass, though.
Thank you. So the next question is from Dot Kelly. It says, I'm concerned that mussels are going to disappear very rapidly as we've seen. Not sure it's been proven what's caused it, but similar to the lobster die-off in Long Island Sound, it is dramatic.
Yeah, and this has been in the news here locally in Maine. If you read the on the
Working
Waterfront, which is a local paper, they fielded a question to a bunch of marine scientists about the mussel problem here, and we contributed a few comments to that. But certainly in their title, up here in the north in the Gulf of Maine, mussels have been on the decline and are hard to find now in many habitats. But we also know that subtitly there's a lot of mussels out there. And there's some folks like Phil Yund at the Down East Institute has actually said that the intertidal in the north, at least, is more of a marginal habitat for mussels. And so we seem to have pretty decent populations in the subtidal, but it is certainly a cause for concern to see those rapid changes that were published recently. I believe I think that's the last 30 years.
I also think you're mapping some of those changes on top, some of these dramatic environmental changes on top of already dynamic evolutionary history, where you have these boom-bust cycles in terms of recruitment. So that, makes it a more complicated system.
Yeah.
Thank you. So I got a follow-up question from Joe Salisbury that asked, what are your thoughts on the reasons for this decline?
Yeah, great question, Joe. And we could talk about it later too on our conference call. But green crabs have certainly been implicated. So top-down control because of invasive green crabs. That effect to me seems certainly spatially variable in terms of what I see in the intertidal green crabs. The water quality itself, as you know, is changing a bit in shallow water. So maybe, you know, a combination of both physiological as well as changes in the kinds of consumers that are out there in the intertidal. But, you know, it's a great scientific question.
Thank you. So I have one last comment or question here from Ashley Taylor that requested, would it be possible to show the final takeaway result slide?
Yeah. That's probably a faster way to do this. This one or the next one? Because I think there's two.
There's also the,
Yeah.
is this what she's interested in? Or is it the bullets?
I'm not sure. I was waiting to see if Ashley would. Ashley said the previous slide, please. Thank you.
There we go.
So I have another question here from Scott Lindell. What are the prospects for producing mussel seed via hatcheries for farming with selected traits and the implications for wild populations?
So it's possible. We can do a fairly simpler genetic screen to find these variants in natural populations, and then we can either use traditional breeding techniques, because actually, you know, you can cross mussels and raise them up. They're fairly fast growers. To bring those genes into something more like a genetic line, or, you know, if you want to do it high tech, you can use this newer so-called CRISPR technology to try to move these genes into the genomes of a target organism. The only issue there is the assumption would be that there'd only be a few genes or one that really cause most of the effect in phenotypes. So a few number of loci with large effects, that works best for this CRISPR technology. If it's got 5 or 6 or maybe 8 or 10 genes of smaller effects, then, you know, using that kind of newer technology is going to be less helpful than the traditional methods of crossing these guys. But I think there's a lot of potential to basically harness this variation from natural populations.
Thank you. At the moment, I'm not seeing any other questions.
Okay.
Oh,
All right.
Oh, we got one more. Sorry. This one is from Melissa Paspini. You mentioned that the phenotype is driven by a handful of rare variants and more common in edulis. How many individuals have this genotype, and are they all heterozygous? Do you think the variants exist due to adaptation or exemption, sorry, my thing moved, of random genetic variation? What is the practical interpretation of an effect size of 8?
So depending on which SNP you look at, there's somewhere between 1 and 5 or 6 mussels that exhibit that rare variant. And what's the second part of that?
The effect size, the practical implications of the quantitative effect size?
Yeah.
18.
8. The effect size? Yeah, I don't know if you can really say 8 has any meaning in and of its, you know, all by itself, but it's in comparison to the other effect sizes, and also, you know, usually when you have an effect size that's, you know, maybe an order of magnitude greater than that, that's usually what you see when you have like these plumage characteristics in birds or whatever, where you have one locus that makes their head yellow or something like that. You'll see effect sizes like 30 or 50. So it's really sort of, it's probably not a very satisfying answer, but it's like, it's Goldilocks, it's like medium.
Thank you. So I have another question from Dot Kelly, who also said, thanks very much for the presentation. Did the higher calcifiers in stress seem regular or like trees, quick growth seems weaker?
I didn't catch that because our facilities is driving by with a lawnmower.
Can you just repeat it?
Sure. Did the higher calcifiers in stress seem regular or like trees, quick growth seems weaker?
Oh, in terms of the shell
Right.
strength, I don't know that we can answer that question because we didn't. We do have all these shells in a freezer, so
Right.
we could test that. But as far as what we measured for our phenotype, we don't have any quantitative measurement of shell
Right.
strength.
But we could go in with those shells and look more closely at shell chemistry, or even a brittleness measure to kind of answer. But that's a great question.
Thank you. And again, I'm not seeing any more questions at this point. So Dwight, did you want to wrap it up for us, please?
Oh, I think you just did. But I just wanted to say thanks very much. It was a fantastic presentation. And I believe this will be posted to the NECAN website at some later point, the saved one. So thank you all very much, and join us next time.
Okay, thank you.
Thanks.